Thanks. So can everyone see my screen fine?
Yes.
Great. So my name is Taylor Fields. I'm the Senior Vice President of IntraBio, and I've been with the company essentially from its very beginning. IntraBio is still a very small biotech company based in Oxford and London. So I'm saying hi from across the pond right now. And our pipeline focuses on developing small molecule drugs for rare and common neurological disorders. Our scientific founders are from the University of Oxford and the University of Munich, but we work with research institutions all over the world. Our lead drug candidate, which I'm going to discuss today, is N-acetylleucine, which is a modified amino acid. And I want to spend a minute just describing its chemical structure so that it elucidates any questions there may be. Amino acids are actually, they can be compounded really as 3 different drugs. And the best way I can describe this if you think about your hands. So N-acetylleucine can be formed as a racemate, a mixture of a D and L enantiomer, which is essentially a 50-50 mix of mirror images. So if you think about your hands, you've got a left hand, a right hand. They look almost identical. They maybe can do similar things, but they actually are fundamentally different. And so that's your L enantiomer and your D enantiomer. And you can also cross your hands, and that's the equivalent of a racemic mixture in a drug form, or N-acetyl D-L-leucine. This racemic mixture, that mixture of the 2 hands, was approved in France in the 1950s under the trade name Tanganil. And so if you've heard Tanganil before, that's again the mix of these 2 L and D enantiomers. It has a very well-established safety profile. It was only approved for the treatment of acute vertigo. And about 10 years ago, one of our founding scientists had the idea that based on what was then known about the molecule, it could be used as a treatment for some of his patients with ataxia. I'll spare the long story there, but in essence, they observed it very rapidly improved symptoms and later noticed that it had a disease-modified neuroprotective effect. And we've now had compassionate use programs in over 20 different neurological disorders, including GM2 gangliosidosis, so Tay-Sachs and Sandhoff. The drug that we're now developing is actually, I'll go back one slide, that L enantiomer, so just the individual L hand. And it's because we've, in all of our non-clinical studies, now realized that the individual L enantiomer, when you take out that D aspect, is more effective, it's safer, and it is more successful in both its neuroprotective and disease-modifying effects. We call this drug IB1001, very clever, I know. And this drug is not currently available anywhere in the world for any condition. It's orally administered. It's transported across the blood-brain barrier, so it works directly on the brain and the cerebellum, but it's also transported to different major organ systems and major organs like the liver and spleen, where we know it has an effect. And again, we believe the drug has broad applicability for both the symptomatic as well as the neuroprotective treatment of diseases like GM2. So when you start off any development program, like you've seen earlier, you generate various evidence along the way to support your rationale. And so we've done a number of animal studies. The one I'm going to show you now is in the GM2 or Sandhoff mouse model. And the mouse on the left is untreated. The mouse on the right has been treated. And you can see that not only can the mouse on the right complete the test, but it's got upper extremity function, hind limb control, a lot more strength, coordination. So this was some of the early evidence that we had, again, that the drug had an effect for these for GM2, and we then sought to translate to the clinic. So on this slide here, it's actually a case study from one of the earliest GM2 patients treated, and it comes from us from the Hand in Hand organization in Germany, who provided this case study, has been a phenomenal partner, and is essentially the NTSAD in Germany. But you'll see on the left here, this is a patient who was untreated, and on the right, you see the patient treated after 9 weeks. Again, the patient still has impairment, but there's a noticeable functional improvement from going to walking with aid to walking independently. And this is just a good example of the rapid improvement that we see with this drug. So I'm going through all this quickly so I can now highlight where we are with the development. Based on all of that evidence, we sought to conduct a multinational clinical trial that would demonstrate the efficacy of the treatment in a controlled clinical setting. And so our trial design first looked at the drug's effect after 6 weeks, so the symptomatic effect, followed by a 6-week washout. And then we have an ongoing extension phase of 2 years where we're looking at the neuroprotective disease modifying effects. The drug was run as an open label study, but we had a novel primary endpoint we utilized, which actually allowed us to use videos of the patient at each visit and allowed for centralized independent raters to make blinded randomized review. So we're able to introduce a high degree of control there. In the US, the study was run for patients 18 and older, but in Europe, the study was run for patients 6 years and older, and it's been a fairly even divide in regards to pediatric and adult patients. So we're intending to continue developing the drug for all ages. I think in our current trial, we've recruited patients between the age of 6 to 64, so a really broad age range. Where we are with this study, we've essentially completed recruitment, but due to COVID-19, the activities have been significantly disrupted. So if you can all think back to the various lockdowns we had, patients were not able to go in for study visits. We had to keep patients on drug for a lot longer than they planned 6 weeks, or the washout for a lot longer than the planned 6 weeks. And we've now had all but one patient complete all of their visits. We're waiting for our study team to be able to go to sites, do what's called source document verification. So making sure that everything entered into an electronic system matches up with the paperwork. And that's something that's absolutely mandatory in order for quality control of your data. And we hope to be able to have the results of the GM2 study within the next 3 months. So I'll of course keep everyone in the community updated on that. What I can share now are the results of an identical study we're running, but for a different indication, which Dr. Walkley actually discussed earlier, and that's Niemann-Pick disease type C. So NPC is also an inherited lysosomal storage disorder. It has a very similar clinical presentation to GM2. And we ran the same trial design, same endpoints with the drug for NPC as we did for GM2. We are very pleased to announce that the NPC study was very successful. Its primary endpoint was met, the secondary endpoints were met, and the drug had was very well tolerated, no serious adverse reactions. And so what you see here are a bunch of strange graphs, but if you look at the boxes in blue, these are how patients change during the treatment period. And the boxes there indicate improvement. And then if you look at the boxes in orange, those are the scores during the washout period. So when patients stop taking the drug, and you can see a deterioration. And these different endpoints represent different things. They're ataxia rating scales, they're clinical global impression scales, they're specific functional index. So it's capturing a whole range of symptoms, functioning, quality of life. And again, the consistency of the data demonstrated a very clear, statistically significant, clinically meaningful effect of the drug, which we're of course hoping that we'll also see when the GM2 analysis is done. That's my presentation for you. I've put up this trial sites here that are participating, although again, at the moment, I don't believe any are recruiting, but I wanted to make sure there was time for questions if there are any, so thank you.
So there's one question that came in about I guess the original racemic mixture versus your more purified drug with the L form, and whether there have been preclinical studies done showing a difference either in efficacy or safety between the two.
Absolutely. So those are actually all published now. The majority of those have come from Professor Francis Platt, who's a scientific founder, but we've now for the last 3 years examined almost every single marker in both the NPC and GM2 mouse model that you could, comparing the racemate, the D and the L, both in regards to their pharmacokinetic, pharmacodynamics, and primary pharmacology. And it's this comprehensive evidence that we have now that demonstrate the mechanism is actually quite unique, but again, that the L is overwhelmingly the effect of enantiomer, that the D is actually quite antagonistic in regards to both biochemical, but also primary pharmacological effects for neuroprotection and disease modification, and namely that the D's pharmacokinetic profile is one that it accumulates, and those long-term accumulations may have toxic effects. So that's all publicly available. Fran and our team have many publications, and again, you can find those on our website.
I have a question of my own. How quickly do you notice effects with this treatment? It seems like the trials are pretty short and you're seeing very rapid onset of changes.
Yeah, so it varies patient by patient. Again, some patients see change in 2 weeks, some patients see a change in 6 weeks, but usually 6 weeks is where you see a clear symptomatic benefit. What we do know now, and this is probably for another day, but that the mechanism for the symptomatic changes and the mechanism for the neuroprotective disease modifying are different. And the way we know that is because we actually see in the animal models that the D enantiomer has this ataxic function for symptomatic treatment. So we'll look at the mice when they're 8 weeks, this is an NPC, when they're at 8 weeks, they're symptomatic, treat for 1 week. At 9 weeks, the L, the DL, and the D all have the symptomatic function. But when you look at pre-symptomatic treatment in the animals, the D again has this antagonistic effect. So if it were the same mechanism, we know that the drug should be doing the same thing. So that's an interesting aspect of it. But for the symptomatic change, I would say 4 to 6 weeks is where we start to see benefit. But we do see in some patients that, again, we believe the drug has a disease modifying effect. So there are patients who after 1 year, we've seen an improvement in their condition. That wasn't the rapid change, but stabilization and prevention of neuronal progression.
So your GM2 study is wrapping up now or soon, and do you plan to approach both the EMA as well as FDA eventually for approval?
Absolutely. So right now we're engaging for MPC because it was done a few months earlier. So we're in conversation with regulators both in Europe, the UK now as it's a different system, and the US about what the next steps are. And if it requires another trial, if it'll be a basket trial, if it can be accelerated approval pathways. So we're having those conversations now.
Great. And the number of diseases that you think this one drug might be useful for?
Well, I'll say the one thing is that as many diseases we found it worked in, we found twice as many that it doesn't. So we don't believe it's snake oil, but we think that the drug predominantly corrects neuronal dysfunction in the cerebellum. I always say it's not a cure. It's not fixing a genetic mutation. It's not unfolding a protein. But as you'll actually, if you look at some of those publications, you can see it's correcting it's common processes of degeneration. So normalizing neuronal membrane potential, mediating neuroinflammation. Perhaps the most notable is we see that it switches ATP production on. So it's increasing mitochondrial function, and it's also normalizing lysosomal volume. So we think, again, for different diseases, there'll be overlapping things. One of the reasons we think it's probably one of the most efficacious for NPC and GM2 is that all of these things are hallmark symptoms of the disease. So we think that's where you're going to see very clear benefit. But we are looking at other ataxias, dementias, other neurological disorders as well.
Great, thanks. And maybe just one last question. Being a rare disease, I suppose we can anticipate that this will not be an inexpensive drug. Any thoughts about pricing and how it compares to Tanganil, which is already on the market?
Sure. So I'll say two things. One, haven't done any pricing yet. We're still trying to figure out if we need another clinical trial or how to get it to the point of approval. But two, and I get asked this question a lot, and I see a question there from another presenter actually, but that everyone who's developing a drug knows that if it's not approved, it doesn't exist. It's technically illegal to import. We've heard stories time and time again about it being stopped at the border. The FDA, we know, is being really serious about cracking down because it is an illegal substance that's unapproved for this indication, both for this indication in France, but also outside the world. And the thing I bring up whenever I'm asked about drug repurposing is that unless it's approved for the indication, it does not exist. There are a whole host of reasons that the company that currently has Tanganil could stop making it, namely because it's been found repeatedly to be ineffective for vertigo. And so if the drug's gone, it's gone. If it's not an approved drug, then you are using an unlicensed agent, what Dr. Toro presented on, and you're not able to monitor it. Your physician isn't able to look at the safety and efficacy. We're not able to track it. We're not able to know if any of the adverse reactions are associated, are related, or what happens. So I would hope that every doctor or scientist on this presentation will want patients to be on approved regulated therapies so that they're getting the best, most managed safe care.
Great. Thanks so much, Taylor. Really interesting data and could be a very useful